Okay, hello. I'm Jessie Kennedy, and I'm with the marketing group at Dovetail Genomics, and I have the pleasure of introducing our speakers today. Thanks for joining us for this webinar. We're going to talk about accelerating your genome assembly projects with the Dovetail AssemblyLink Assay and some of the innovations we've been making at Dovetail. Today, our speakers are going to be Dr. Miriam Elkawand. She's our head of customer success at Dovetail, and Billie Maguire. He's a Ph.D. student in the Vidal Lab at UMass Boston. Miriam leads customer success team at Dovetail Genomics, and she's been with the company for 6 years, helping customers achieve their research goals using the Dovetail proximity ligation kits and analysis solutions. Today, Miriam's going to walk us through the innovations that we've been making at Dovetail with the AssemblyLink Assay, explaining how you can generate Hi-C data for your genome assembly project in just a single day. Billie is a Ph.D. student in the Vidal Lab at the University of Massachusetts, and he's studying ecological genomics of Hyphantria cunea, or fall webworm. Billie graduated with an undergraduate degree from Georgetown University in biology, and he studied ecology and genetics of interacting plants and insects in salt marshes there. Today, Billie's going to walk us through the contributions of population genetics, genome structure, and genetic architecture, and phenotypic plasticity to dietary generalism in insect herbivores. He's going to showcase the importance of having a high-quality genome assembly for achieving high-resolution data, reducing sequencing costs, and enhancing confidence in variant calling, all of which are essential for these eco-evolutionary studies. So we're very pleased to have Billie here with us today. And all right, without further ado, I'm going to hand it over to Miriam to kick us off with a nice introduction.
Well, thanks everybody for joining. In today's presentation, I'll walk you through the Dovetail AssemblyLink Assay and how it can accelerate your genome assembly projects. So to start with, I'll tell you a little bit about Dovetail Genomics and who we are. Then for those of you who are not familiar with the Hi-C proximity ligation technique, which is the core technique that our assays are built on and have evolved from, I'll tell you what Hi-C is and how it contributes to genome assembly. Then I'll spend the rest of the slides walking you through the AssemblyLink Assay, which is the latest assay for genome assembly applications launched by Dovetail Genomics. Dovetail Genomics is a leading brand in genome assembly. The company was founded in 2013 and has since delivered over 2,000 assembled genomes of varying sizes, so a few hundred megabases to over 30 gigabases. There are more than 600 articles published with our technologies, and the species that have been worked on span the entire tree of life. We offer kits for researchers who wish to run the assays in their lab. We also offer services, both wet lab or library prep services, as well as informatics or data analysis services. So core to what we do is the Hi-C proximity ligation technique. Despite the improvements that have been done with the DNA sequencing technologies, such as the introduction of PacBio long-read sequencing, this data by itself often does not generate a chromosome-level assembly. So what you end up with is a draft assembly made of several contigs or scaffolds that are considerably shorter than full chromosomes. And this is where Hi-C comes in. Hi-C data bridges the gap between a draft assembly and a chromosome-level assembly. So what is Hi-C? Hi-C captures interactions between different genomic regions. So what you do is first you cross-link the chromatin in the nucleus using standard cross-linkers such as formaldehyde. Then you fragment the chromatin via an enzymatic reaction, and you ligate fragments that are in close proximity. So what you end up with is this chimeric DNA molecules that is linking two separate genomic regions that were held in close proximity in the 3D space without potentially being next to each other in the linear sequence space. After the proximity ligation, you reverse the crosslinks, degrade the proteins, and you purify the DNA. Then this DNA is converted into a sequenceable library, and the library is sequenced in paired end mode. So the read pairs that you end up with have one read that is aligning to one genomic region, and the other lead aligning to another separate genomic region. And the linear distance between the genomic regions can span single base pairs to megabases and up to full chromosomes. So what you're looking at here is this dot that represents a contact between two specific genomic regions that you're able to locate and position. So in this example, this contact is between this region and this region. And in this example, the contact is between this region and this region of the genome. So one way to visualize Hi-C read pairs is through a contact map, where the contact represents, or the dot represents a contact between two specific regions, as I just mentioned, but also the intensity of the color represents the frequency of the contact. So the darker the color, the more contacts. The lighter the color, the less contacts. There are two things I would like you to take from this contact map. One is that chromosomes don't interact much with each other. That's why you have less contacts in this trend space compared to more contacts as evidenced by this intense color within chromosomes. Two, regions that are located close to each other in the linear sequence tend to interact more frequently with each other. So the closer you are to this baseline, the more interaction you have, which is why you have this more contacts and more dark color. The more you move away from this baseline, the more the linear distance between genomic regions increases and the less contacts you have. And this is the information that is provided by Hi-C data that allows you to order and orient contigs to increase the contiguity of the assembly up to a chromosome level. So in this cartoon, I'm trying to illustrate exactly that, how Hi-C enables the ordering and the orientation of contigs from your draft assembly. So you start with your draft assembly. This could be your PacBio Hi-Fi assembly. In this example, you have 4 contigs. You obviously have several more, but for this example, we're just having contig A, contig B, contig C, and contig D. You generate your Hi-C data, you analyze them, and what you see is that you have a lot of contacts between A and C, which is why you have this brighter red, and you have less contacts between A and B, and very few contacts between A and D. So what you can infer from this is D likely belongs to a separate chromosome, a different chromosome, and C is actually closer to A than B. So the right ordering is A, C, B, then D. Using the same Hi-C data, you're able to see that A, or this right end of contig A, has more contacts with the right end of contig C and less contacts with the left end of contig C. So contig C is actually in the opposite orientation. So that's how Hi-C is allowing you to order and orient contigs and allowing you to increase the contiguity of your draft assembly up to a chromosome level. But the question is, if Hi-C is needed in genome assembly, why it's not always used, or why there are barriers to its adoption by the community still? The reason for that are mainly 2 things. One is existing Hi-C workflows are long and laborious. So they have complex steps, and it takes 2, 3, or more days to complete a Hi-C workflow. 2, the existing Hi-C workflows use restriction enzymes to digest the chromatin. So you're really limited to where you have a restriction enzyme site. And parts of the genome that has less or don't have a lot of restriction enzyme sites, you're blind to them. So there are gaps in the data that the existing Hi-C workflow is generating. And that's what we're trying to solve for with the launch of the AssemblyLink assay. The AssemblyLink assay is based on the link prep chemistry that is recently launched by Dovetail Genomics, and it's a TN5-based Hi-C. So the AssemblyLink workflow shares similarities with the workflow, the Hi-C workflow that I previously discussed, but also has key changes that help solve the challenges of existing Hi-C methods. And I'll walk you through these changes. You start with your sample. It can be blood, tissue, or cell. And then you cross-link the chromatin in the nucleus. The difference here is we don't only use formaldehyde for cross-linking. we use dual cross-linkers. We use formaldehyde and we use DSG. So formaldehyde cross-links DNA to proteins and proteins to proteins that are close to each other. DSG has a longer spacer arm, and it's a strong protein-to-protein cross-linkers, so it allows to cross-link proteins to proteins that are more distant apart. And by doing so, by using DSG and formaldehyde, we're increasing the long-range information that is captured in your libraries. Then we move on to fragmenting the chromatin, and here we use TN5, so we don't use a restriction enzyme. And TN5 is sequence agnostic. So unlike a restriction enzyme, it's not biased by a specific site on the genome. So what it allows you to do is it allows you to have an even fragmentation of the chromatin that results in a uniform coverage and an unbiased coverage. After fragmentation, you set up the proximity ligation reactions. You end up with a proximity ligated DNA. That's again bringing together regions of the genome that are in close proximity. You purify the DNA and you convert it into a sequenceable library. And here for the library conversion, you don't need to do sonication and you don't need to do biotin pull down. The assay leverages pre-tagged adapters that are added during the fragmentation step. So what that means is you're able to speed up the process. You're removing the need of a sonicator, you're reducing variability that comes with that step, and you're also reducing the number of invalid read pairs in your Hi-C library. So the main implication for AssemblyLink assay is the speed that it offers. It allows customers to go from sample to sequencing ready library in a single day. That's less than half the time that is needed for traditional Hi-C assays. And AssemblyLink assay is the only assay on the market that offers the ability to start your prep in the morning, and by the end of the day, have your libraries ready to put on the sequencer. In addition to speed, I'd like to highlight other benefits for the AssemblyLink assay. It's simple to use, and it is accessible to novice Hi-C users. It is compatible with multiple sample types, cell lines, blood, animal tissues, plant leaves. It supports reduced input requirements down to 5 milligrams of tissue. It is easy to automate if you wish to automate it. And it integrates easily with existing Hi-C data analysis workflows, meaning you don't have to re-engineer the Hi-C scaffolding pipelines that the lab is currently using. One thing to stress on is the AssemblyLink offers all of these benefits without compromising on the quality. So the AssemblyLink assay delivers high-quality Hi-C libraries as assessed by standard Hi-C library metrics. What we're looking at here is a library prepared from GM12878 cells using the AssemblyLink assay and using comparator assays that use restriction enzyme, that are restriction enzyme based. The libraries were downsampled to 1 million read pairs and were processed through standard tools that are used in the community or in the field to process Hi-C data, namely BWA aligner and pair tools. And what we're looking at is the standard QC metrics for Hi-C libraries, which is the percentage of mapped read pairs, percentage of no-do, percentage of trans and cis, and the percentage of long-range cis read pairs. And what you can see is that the AssemblyLink library is enriched in long-range information, which is the hallmark of Hi-C assays. It also has a higher percentage of mapped read pairs and a higher percentage of cis read pairs. And this results in more informative read pairs for the downstream analysis. This is a view into the different categories of samples that the assay supports and the minimal requirements that are needed. So the standard workflow for mammalian cells calls for 1 million cells, 3 ml for mammalian blood, 5 milligram to 20 milligram for animal tissues. And the animal tissues represents mammals, birds, fish, insects, marine invertebrates, so a wide variety of animals. And then plant leaves, 30 milligram. We're very excited about these minimal requirements. So as comparison, competitor assays would require 50 to 200 milligram of tissues and around 1 gram of plant leaves. And the reason we're very excited about these minimal tissues is that, or minimal requirements, is that we believe it will enable researchers to actually work with samples with limited material or small specimens. And this excitement is echoed by Minderoo Oceanomics Center at the University of Western Australia. They're working towards creating a library of high-quality reference assemblies for marine vertebrate species, and they shared that the reduced input requirements from fresh frozen animal tissues means that they can now tackle even the smallest specimen. So the high-quality libraries that is generated by the AssemblyLink assay supports high-quality assemblies. And what you're looking at here are the stats of an assembly that was generated by scaffolding a PacBio Hi-Fi draft assembly from a plant sample with AssemblyLink data. And what you can see is a significant improvement in the N50 and L50 in the final assembly that was scaffolded with AssemblyLink compared to the draft assembly. You also have a high BUSCO score. So all of these stats are consistent with high contiguity and highly complete assembly. For those of you in the audience who are familiar with the Dovetail Omni-C assay, which was the first restriction enzyme-free high-C assay for genome assembly that was launched by Dovetail Genomics a few years back, you may be wondering how the scaffolding performance of AssemblyLink compares with Omni-C. And we've looked at that across several samples during the development phase, and we found that the scaffolding performance of AssemblyLink is highly comparable to Omni-C. So what we're looking at here is a synteny plot comparing this assembly that was scaffolded with AssemblyLink with an assembly that was generated from the same PacBio draft assembly, but scaffolded with Omni-C instead. And you can see that there is a perfect alignment of the large scaffolds. So with AssemblyLink assay, you're getting the same quality, the same high quality assemblies, but with a shorter and more streamlined workflow. What you're looking at here is a pseudohaploid assembly, meaning that the two parental haplotypes were collapsed into a single version. So in your final assembly, you have one haplotype at heterozygous loci. What the community has been working towards now, and this is picking up more and more, is haplotype-resolved assembly, where you actually have two independent assemblies of the parental chromosomes, and that is the true representation of your sample genome. AssemblyLink, because of the uniform coverage, enables you to generate high-quality haplotype-resolved assembly because it enables you to have a more comprehensive detection of heterozygous SNPs and the ability to phase them across the chromosome. So here we're showing libraries that generated from AssemblyLink and competitor assays from the same sample sequenced to 800 million read pairs, so same depth and aligned. And what you see is the traditional high-C methods or traditional high-C libraries that are using restriction enzymes have this wide distribution. That's here in blue and in this orange. And also they have parts of the genomes which are under-sequenced and underrepresented. And that's because these regions of the genome would have few or no restriction enzyme sites. AssemblyLink distribution shows this shotgun-like uniform coverage distribution, and that's what enables you to have a higher number of SNPs or heterozygous SNPs that are detected, and then a high accuracy in phasing those SNPs across the chromosomes, and that results in high-quality haplotype-resolved assemblies generated or enabled by AssemblyLink data. And that's an example that we're showing two assembled haplotypes of one fish diploid genome. And these haplotypes or these assemblies were enabled by AssemblyLink data. And what you see is high contiguity of both assemblies. You don't see off-diagonal signal that would be indicative of misassembly. So you have high accurate assemblies that's also evidenced by these numbers. So what we're showing here is a high BUSCO score for high completeness and a high Mercury score for both haplotypes, indicative of also high accuracy. So we're currently offering the AssemblyLink assay as a kit. The kit supports 8 reactions. It's an end-to-end reaction kit, so there is no need to purchase additional library preparation kits. It offers flexibility for various genome sizes, so you're able to split the proximity ligated DNA into multiple libraries if you're working with large genome sizes, so more than 3 gigabases. You don't have to repeat the workflow from the beginning. You can use your proximity ligated DNA and convert that into multiple libraries to support the depth that you need for your genome size. The kit is also accompanied by a detailed guidance on library QC, so you're able to assess the library of the quality of the library you're generating based on, again, standard high CQC metrics. The data is compatible with open source scaffolders such as Yaz and Salsa2. Alternatively, Dovetail also offers a scaffolding service, high-rise scaffolder pipeline, that is also compatible with the AssemblyLink data. And I've added to this slide the QR code for the product page if you wish to learn a little bit more about the AssemblyLink assay and kit. So to close off, I hope I was able to convey to you why we're excited about the Dovetail AssemblyLink assay and why we think that this is a next generation Hi-C assay for genome assembly. Again, to summarize, it enables sample to sequence ready library in a single day. It supports a broad assortment of sample types, requires minimal input. It empowers genome scaffolding, haplotype-resolved assembly, and polyploid assembly as well. With that, I'll stop sharing and I'll hand it over to Billie to take it from here.
Okay. Hi, everyone. Thank you, Miriam. My name is Billie Maguire, and here's a picture of me out in the field doing some field work, collecting my fall webworm caterpillars from up in the trees. But today I'm not in the field. I'm here talking to you about the genomics of dietary generalism and the use cases for a haplotype-resolved assembly in this type of research and in eco-evolution research more broadly. So first, a little bit about what motivates my research. I'm interested in the diversity of life on Earth, particularly eukaryotic diversity. So that's mostly the diversity of plants, animals, and fungi. And if you start studying eukaryotic diversity for long enough, one of the first things that you'll notice is that the vast majority, or the majority at least, of eukaryotes are insects. And so if we want to understand this diversity of eukaryotic life, all this diversity of function and form, studying the diversity of insects is a good place to start. And if you start studying insect diversity for long enough, one of the first things you'll notice is that most insects are herbivores. That represents over 470,000 species, and most insect herbivores are highly specialized. So I've tried to illustrate that with the histogram that I borrowed from Forrester et al. Here. And what this shows is that the vast majority of insect herbivores are feeding on only a single plant family. And then there's this very long, thin tail of highly generalized species. So there's many, many specialists and very few generalists. And so we think that this specialization is related to insect diversity. And this is the problem that I'm really interested in, which is the relative diversity of specialization and the relative paucity or rarity of generalization. What's driving these patterns? And the system that I use to address this is fall webworm. Its Latin name is Hyphantria cunea, and it's an rabid moth. It's an extremely generalized moth, so it's way out on the tail of this distribution here that I showed you previously, feeding over 600 species of plants. And I've included a picture of the caterpillar here. It lives gregariously in these colonies with its siblings in webs high up in the trees where it eats the leaves. And what's interesting about this caterpillar is that it has a very wide range. So I've included a map from iNaturalist where each orange square represents an observation of fall webworm. And as you can see, fall webworm really dominates the eastern half of the United States, but its range really spans from the Atlantic to the Pacific and from Mexico to Canada. And throughout its very large range, it's encountering a number of different hosts throughout its range. So it's eating and encountering different things in Florida than it is in Maine, than it is in California, than it is in Canada. And so its diet breadth has this geographic pattern. Its diet varies locally. Another thing that's interesting about fall webworm is that it's composed of 2 biotypes, the blackhead and the redhead. So the black head is shown here and the red head over here. And these are clearly morphologically distinguishable by the color of their head capsules. And they're also highly genetically distinct. So they're very highly genetically diverged. And what's interesting about these two biotypes is that the blackhead has a much larger diet. It eats more plants than the redhead, and the redhead eats fewer plants relative to the blackhead. And so this makes fall webworm an attractive model system for studying dietary generalism because we have dietary generalism or diet that varies along these two axes, this geographic and local axis, and also this genetic axis. And this allows us to ask some questions about what contributes to dietary generalism, about what some of the genetic and genomic contributions to dietary generalism are. And so one thing we can think about is population genetics, and we can ask whether generalists are locally adapted, and we can address this with population genetic studies across geography. Next, we can ask if the genome structure or genetic architecture of generalists are different. Do generalists have a different genome structure? And we can address this with long-read sequencing to detect structural variants. And finally, we want to know about phenotypic plasticity. Are generalists more plastic? Are they able to modify their genomes epigenetically in certain ways that allow them to feed on certain plants? And we can address this with methylome sequencing with nanopore. And so these are three areas of research that are ongoing in the lab, and they also represent three chapters of my dissertation research. So I don't have too many results to present to you today, but really what I want to try to do is give you a little bit of background on this research, explain why it's important, and kind of give you an idea of why a haplotype-resolved assembly is really essential for doing any of this research. And so we needed a haplotype assembly for fall webworm, and we thought we had a high-quality assembly because Wu et al. Published a fall webworm genome, and they made it publicly available in NCBI. However, this genome was of poor quality. And so I've shown that in this red line here. On the x-axis, we have the percentage of the genome covered, and on the y-axis, we have the scaffold length. As you can see, the Wu et al. Genome here, shown in red, is composed of very, very many, very small scaffolds. So this is a poor quality genome. So we turned to AssemblyLink to generate a much higher quality genome, which is shown here in blue. And as you can see, the vast majority of the genome is covered by these very large scaffolds, indicating a much higher quality assembly. And it's even comparable to the silkworm moth, as you can see here in pink, which is the highest quality genome that we have for a butterfly or moth available, publicly available to science. And so we've generated this very high quality assembly with this AssemblyLink kit. And so the way Miriam talked much more about this, but just briefly, we fully phased PacBio Hi-Fi reads using Hi-C integrated Hi-Fiasm and AssemblyLink data. We then scaffolded the two draft haplotype assembly using Dovetail high-rise scaffolding pipeline, guided by the long-range information captured by AssemblyLink read pairs. And it generated this genome. And so I've included some statistics for each haplotype from the genome, the two haplotype genomes that were generated. And one thing to notice is that the BUSCO score is very high. So that's the universal single copy orthologs. So we have most of the genes, the vast majority of the genes that we would expect to see in fall webworm are here. There's a single fragmented gene, only a single fragmented gene. And we have very little missingness in our data. We have few gaps and we have few ends or missing bases in our data set. Another thing to point out is the L90 is 29. And so that's the number of scaffolds that it takes to cover 90% of the genome. And we know from closely related species that we expect fall webworm to have 31 chromosomes. And so an L90 of 29 indicates to us that this is very near a chromosome level assembly, and that each scaffold likely represents a chromosome. And so we have this very high quality genome assembly now, thanks to AssemblyLink, and we're able to do some research with it. The first one I want to talk about is population genetics. And so a little bit of background on this. One hypothesis for how generalism could be maintained is that generalists are locally adapted. So generalists could be composed of subpopulations that are one, genetically differentiated, and two, locally adapted. And so I've tried to illustrate that with a figure here. In the red, we have an overall generalized population, and this red population is feeding competently on hosts A, B, and C. But in reality, it's composed of two subpopulations, this orange subpopulation and this blue subpopulation. The blue subpopulation is specialized to feed on host C, which is a local host to the blue area. And the orange subpopulation is specialized to feed on host A and B, which are local hosts to the orange area. And so really, when we look at the species level, we see an overall pattern of generalization, but really when we look closer, we see specialization and local adaptation. And so if this were the case, if generalists were really just locally adapted specialists, we would expect to see signatures of divergent selection across its range. And so we can test that with some genomic data, but first we have to do some sampling. So we sampled fall webworm from these locations across the United States. The purple triangles represent places we sampled from this summer, but we didn't have a chance to include those dots on the map yet. And each dot represents an individual caterpillar that we sampled from the wild and sequenced the genome for. The red dots are the red-headed caterpillars, and the black dots are the black-headed caterpillars. And so we generated whole genome data from these individuals, and we can look at single nucleotide polymorphisms to try to look at signatures of divergent selection across the range, which might be signatures of local adaptation and local specialization that could be driving dietary generalism in our species. And there are a couple of different ways to getting this sort of data to try to understand divergent selection across the genome. One approach that we could take is RAD-seq. This is a reduced representation approach where we sequence a subset of the genome. We chop the genome into pieces and sequence a subset of those pieces and get high coverage for all of those pieces. And this is advantageous because we are able to achieve high coverage. However, the downside is that we're losing important information because we're not sequencing the whole genome. So we might be losing functional regions of the genome that could be important in driving divergent selection. So we opted not to go with this method. Another possibility is to go with pool-seq, which is where you pool all the samples into a single library and sequence it all in one lane without barcodes. And this allows you to retain whole genome information at high coverage. However, you don't have any specificity. You can't identify particular SNPs that are associated with particular individuals. So we decided not to go with this option either. So instead, we decided to go for this third option here, which is called low coverage whole genome sequencing. And in this method, we sequence the entire genome of every individual, but at low coverage. So this is advantageous because we retain whole genome information and we retain that individual level information and that individual level specificity. However, there can be problems because there's missingness and low coverage in our data set, which makes having a haplotype-resolved assembly especially important. So a haplotype-resolved assembly enables, in this case, improved reliability of variant detection in already sparse data. A phased haplotype assembly helps reduce errors in variant calling, particularly when detecting heterozygous variants. They provide a clearer picture of whether mutations are real or due to sequencing or assembly artifacts. It also reduces data loss through efficient and accurate mapping. So in a pseudo-haplotype assembly, you might have regions of the genome that are duplicated or missing. And so your reads map twice, or they overmap, or they undermap, or they don't map at all to the genome. And so we're losing data in this case. And when this is particularly bad because our data is already sparse. And so together, these two things allow us to do whole genome, a haplotype-resolved genome assembly allows us to do low coverage whole genome sequencing, which allows us to save costs on sequencing effort and cost savings. Next, I want to talk about genome structure and genetic architecture. And we can think here about how generalists might have a different genetic structure, like large-scale differences in the genome that could be contributing to dietary generalism. And we can adjust this with long-read sequencing for structural variants. So a little bit of background on structural variant detection. Structural variants are a widespread type of mutation of 50 base pairs or greater. So now we're talking about much larger mutations instead of the single point mutations that we were talking about earlier. And these types of mutations can have large fitness effects. And they can also contribute to reproductive isolation and speciation, which are both important processes in the generation of biotic diversity. And particularly, we're interested in duplication and transposable element insertions. These are two common types of structural variants across the tree of life, but they are particularly important in insects because they've been linked to the tolerance of xenobiotics, which include pesticides and also plant secondary compounds that the plants use to deter herbivory. And so we want to look at these mutations in addition to SNPs. And so here's a little figure that tries to show a bit of an outline of how I plan to do this. The first step would be to sequence a single redhead individual and a single blackhead individual at high coverage for long read data. And log reads are advantageous for detecting structural variants because the reads are long and they span the entire width of the structural variant. So it allows you to confidently assess structural variants and discover them de novo. However, this approach is not super feasible for assessing the number of structural variants at a population level scale. So if we really want to dig in and see how is the frequency of structural variants varying across populations, we have to turn to short read sequencing. And short reads are less reliable for detecting structural variants because they're short and they often don't span the entire structural variant. But we can use some bioinformatic methods to account for this, and we can talk about this later if you guys have questions. But basically, we use these two approaches together, one to discover the de novo structural variants, and one to discover the frequency of those structural variants in order to get a picture of the overall landscape of structural variants in fall webworm in the species. And so a haplotype-resolved assembly is really important for this research because it enables better resolution of structurally complex regions, which are hotspots for SVs. So the long-range information provided by a haplotype assembly is better for identifying structural variants. These variants are often missed or inaccurately reconstructed in unphased or pseudo-haplotype assemblies. And they're also important for resolving phasing of structural variants in high coverage samples. So a haplotype assembly provides a highly accurate phase genome, which means you know exactly which alleles are linked together on a specific chromosome. And finally, I want to talk about phenotypic plasticity, where we can think about if generalists are more plastic and if they can manipulate their genome through epigenetics in order to make them more tolerant of certain host plants and expand their host range and diet breadth. And we can address this with methylome sequencing. So a little bit of background on epigenetics and methylation. Cytosine methylation is detectable with nanopore with no special library prep, which makes it very easy for us. And methylation can have both a genetic and an environmental basis. So in our system, we might think about the genetic basis as biotype. These biotypes are highly genetically diverged, so we think that they might have different methylation patterns. And they can also vary by environment, which in our system we think about as the host plant. So these caterpillars are feeding on different hosts, which could induce different environmental changes or epigenetic changes to their genome, which could induce plasticity and allow them to feed on different host plants. And so we want to test for genome-wide patterns of methylation with respect to this genetic axis, this biotype axis, and also this environmental or this host plant axis. And we can also test for associations between methylation and performance, between fitness metrics. And so here's a figure that outlines how I plan to do this. In this case, we take maternal lines from blackheads and redheads, as many as we can, because that's a replication in this case. And we take those maternal lines, and we take offspring from those maternal lines, and we mate the siblings together. And from those sibling matings, we take the offspring, and we rear them on two different hosts, and we rear them until they reach pupation, which is the developmental stage between the larvae and the adult. And at this stage, we can measure some important fitness metrics about them, which include development time and pupal mass, which are good indicators of fitness in insect herbivores. And we can also homogenize the tissue, extract the DNA, prep the libraries, and sequence them on nanopore machines to get both base call data and methylation call data. And then we can use some statistical approaches to try to look at overall patterns of genome-wide methylation. So we can use a PCA to see if there's overall differences between host plant or biotype. And we can regress some of these PCA axes against some of these fitness metrics that we have in order to see if overall patterns of methylation are predicting fitness on certain hosts. And so a high quality genome is really important for this research because it allows us to conduct nanopore sequencing at medium coverage. And so nanopore base calls are less reliable than Illumina calls. So having a good quality genome is especially important in this case. And it also allows us to avoid technically challenging and toxic bisulfite sequencing. So we don't have to do this bisulfite sequencing, and we're not having to assemble a de novo bisulfite genome every time we want to sequence a caterpillar or a pupa. Instead, we can rely on nanopore sequencing and our high quality genome instead. So in conclusion, a contiguous annotated haplotype-resolved assembly saves costs on downstream sequencing effort. It enables more confident variant calling with low coverage data. It enables resolution of phasing and high coverage data, and it's absolutely essential for resolving structural variants. And great, and that's the end of my presentation. Thank you very much for your attention, and I'd just like to acknowledge my lab mates, collaborators, advisors, and funding sources, which include Myra Vidal, Brooke Moyers, and Matt Comerford. And I'd like to thank the NSF for funding this research. Thank you very much, and I think now we're open up to questions.